Okay, hi everyone, welcome, and thank you for joining our today's webinar titled The Role of Dietary Lutein and Zeaxanthin in Cognition in Early Life. My name is Haneen Abu Leila, and I am the country coordinator for the Jordan Nutrition Innovation Lab. I am really thrilled to open our second webinar for this year. And as more attendees are joining, I will begin by going over some of the logistical items for our webinar today. Next, please. I would like to direct all attendees to a few functions on this Zoom call. At the bottom of your screen, you should see a chat icon and a Q&A icon. Please use the chat feature to engage in relevant conversations with the other attendees. Next, please. If you have a question for our speaker, please use the Q&A feature. We have allotted the final 15 minutes of this webinar for Q&A, at which point our speaker will respond to any questions from the audience. This webinar is being recorded and will be made available on our website following the event. We will repeat these technical housekeeping items in the chat throughout the webinar as people may be joining in later times. Next, please. Great. So before starting our webinar, I would like to give a small introduction about our project. The Jordan Nutrition Innovation Lab is a research and capacity building program that is funded by USAID Jordan under Feed the Future initiative. We pursue research and capacity building activities to support the health and nutrition of women of reproductive age, pregnant lactating women, and infants and young children in Jordan. Some of our key activities are implementing a rigorous maternal and child nutrition research agenda, including conducting analyses of existing nationally representative data sets and conducting comprehensive evaluations of programs such as USAID's Community Health and Nutrition Program. Moreover, we build individual and institutional capacity through the award of fellowships, scientific symposia, workshops, and webinars. We conduct these webinars on a regular basis as a means to conduct outreach to a wider stakeholder group in and outside Jordan. The webinar is, our webinar today is moderated by Dr. Lynn Osman. Before we start, I would like to introduce Dr. Osman. Dr. Lynn is Saqr bin Muhammad Al-Qasimi Professor in International Nutrition, as well as a professor at the Friedman School of Nutrition, Science and Policy at Tufts University. She has been an active investigator in USAID-funded projects in Malawi, Uganda, Nepal, and most recently in Jordan as part of the Feed the Future Jordan Nutrition Innovation Lab. She has been active in experimental studies with humans on lipoprotein response to several vegetable oils, trans fats, and soy protein, and the glycemic response as modified by various meal components. Most recently, she has been examining the molecular mechanism of action of carotenoids, both vitamin A and non-vitamin A, both pro-vitamin A and non-pro-vitamin A against several chronic diseases. Dr. Lynn, the floor is yours.
Thank you very much, Haneen. Now I have the pleasure of introducing our speaker, Dr. Haya Mamasani. Dr. Haya is a researcher at the Public Health Authority of Saudi Arabia. Her work focuses on leveraging survey and healthcare data to inform public health policies and interventions. Prior to this role, she was a health specialist at the World Bank, where she designed and supported the implementation of health and nutrition projects in countries of the Middle East and North Africa. Dr. Haya obtained her PhD in nutritional epidemiology and an MPH from Tufts University. Dr. Haya, over to you.
Okay. Hello, everyone. Thank you so much, Lynn, Shibani, Maha, and Haneen, and all of the Jordan Nutrition Innovation Lab for the invitation to speak today. Thank you to all the attendees for joining. So I'll be talking today about the role of lutein and zeaxanthin in cognition in early life. Here's an outline of my presentation. I'll start with a brief overview of neurodevelopment, then introduce carotenoids. Then I'll talk about the evidence showing the relationship between lutein and zeaxanthin intake and cognitive function in older age. Then I'll present some emerging evidence of a possible role of these carotenoids in cognition in early life. And then I'll conclude with some future directions, and I'd love to hear from you as well. So I'm starting with a figure of the major developmental milestones of gestation. So pregnancy, gestation may be the most sensitive stage of the life cycle. So during the embryonic phase, that's from conception until week 8, the major organs of the body are formed, such as the central nervous system, the heart, and the digestive system. During the fetal phase, from week 8 until birth, the structures formed in the embryonic phase grow and mature rapidly. And by 38 to 40 weeks, a healthy fetus has built substantial nutrient stores and developed sufficiently to thrive outside of the uterus. Now, specifically, human brain development is a complex process. It begins in the first few weeks after conception and continues postnatally, at least through adolescence and arguably throughout the lifespan. The first key event of brain development is the formation of the neural tube. That's neurulation on the figure. This process is usually complete by 4 weeks after conception. From 4 to 12 weeks, the neural tube differentiates, eventually becoming the brain and the spinal cord. About 7 weeks after conception, the first neurons and the first synapses, that is the connections between the neurons, begin to develop in the spinal cord, and they migrate to different brain areas where they form connections. In the last trimester of pregnancy, myelination, which is the fatty insulation of neurons to allow them to process information quicker, and formation of synapses, that's the connections between the neurons, which are essential for the development of the functions of the brain. And these also continue postnatally. So the different functions of the brain develop and mature at different rates. And the maturation of specific brain areas during childhood is associated with the development of specific cognitive functions. The first brain regions to mature are those involved in visual control, balance, and motor abilities. Then the regions which enable learning, memory, language acquisition. And then lastly, the frontal lobes, which are responsible for higher level cognitive functions, that's self-regulation, problem solving, for example. These are the last brain areas to develop. So nutritional deficiencies and other insults to the brain at various of these stages of gestation, infancy, and childhood are likely to affect brain functions that are developing during that time, which may have long-term cognitive consequences. So my takeaway from these 2 slides is that the most crucial developmental period of the human central nervous system happens early in life. That's during gestation, infancy, and early childhood. Now, brain development is guided by the interplay of genetic and environmental factors, one of which is nutrition. And optimal brain development in the prenatal period and the early years of life depend on providing sufficient quantities of key nutrients during specific sensitive periods. And while all nutrients are important for brain development, certain nutrients such as iron, zinc, iodine, B vitamins have particularly significant effects early in life. Now moving on to carotenoids. Carotenoids are a family of compounds of hundreds of pigments that are synthesized by plants, and they provide much of the color we see in nature. For example, red in tomatoes, orange in carrots, even the autumn leaves that we see, these are carotenoids that are giving them the yellow-orange colors. Humans obtain carotenoids from the diet and accumulate them in tissues and plasma, where they play a variety of biological functions. And a small portion of these carotenoids are considered to be beneficial in the prevention of major diseases such as cancers and eye disease. The carotenoids that have been most studied in relation to health are beta-carotene, lycopene, and lutein and zeaxanthin. And based on their chemical structure, beta-carotene, alpha-carotene, and lycopene belong to a class of carotenoids called carotenes, and lutein and zeaxanthin and beta-cryptoxanthin belong to a class of carotenoids called xanthophylls. Because they contain one hydroxyl group, as you see in the structure, they are more polar. Beta, alpha, and beta-cryptoxanthin are vitamin A precursors, so they can be converted to vitamin A in the body. Vitamin A is an important nutrient for normal vision, the immune function, reproduction, and growth and development. For this presentation, I'm going to be focusing on lutein and zeaxanthin. The major sources of lutein and zeaxanthin are dark green leafy vegetables, corn, orange-yellow fruits, avocados, and egg yolk are highly bioavailable sources. Now, data from food composition databases on lutein content include zeaxanthin, so we cannot kind of examine the effects separately in observational studies. That's why I'm referring to them together. However, in general, they are similar in terms of food sources, human metabolism, and tissue storage. So we would examine dietary lutein and zeaxanthin as one exposure. And carotenoids are best absorbed when they are taken with a meal containing fat. The mean intake of lutein and zeaxanthin in the US is around 1.7 milligrams per day in adults, 0.7 milligrams per day in children. In Jordan, I couldn't find any data on lutein and zeaxanthin intake specifically. If any of the attendees has this information, please do share with us at the end. But there is some indication that the intake is low. The latest National Nutrition Survey in 2019 showed that 44% of children 6 to 23 months old consumed 0 fruits and vegetables in the previous day. In the DHS of 2017, 14% of children consumed dark green leafy vegetables only in the last 24 hours. In adults, 84% of adults consumed less than 5 fruits and vegetable servings per day. So intake seems to be low. There's no recommended intake for lutein and zeaxanthin, but as I'll show you in the next slide, lutein and zeaxanthin are critical for the health of the macula, that's the back of the retina in the eye. And it's been shown that intake of 6 milligrams per day can protect from age-related macular degeneration, which is a common eye disease causing blurring of the central vision. And it's a leading cause of visual impairment and blindness in the US and in many other countries. In the next few slides, I'll present some evidence supporting a role for lutein and zeaxanthin in cognition. Now, although not the major carotenoids in the diet, lutein and zeaxanthin tend to be dominant carotenoids in central nervous system tissues. So think of it like that. Out of the hundreds of carotenoids in nature, there are 50 that are found in the diet, around 2 dozens that are detected in the body. Only lutein and zeaxanthin are found in the macula of the retina, where they form what we call the macular pigment. That's the concentration of lutein and zeaxanthin in the macula. Now, as you can see on the slide, the retina is the layer of the nerve cells in the back of the eyeball. It senses the light and sends signal to the brain so we can make sense of what we are seeing. The macula is a tiny area in the center of the retina, which is responsible for acute central vision. So this is the cross-section here of the macula. So I think of this as lutein and zeaxanthin in the macula, they're believed to protect photoreceptors from blue light and oxidative damage, and therefore they protect from age-related macular degeneration. So I think of the macular pigment kind of like internal sunglasses that protect our photoreceptors from oxidative damage and blue light. So here's a study conducted by Dr. Johnson and colleagues, where they looked at brain carotenoids in centenarians, so 100 years and older, who volunteered to donate their brain upon death. When we comparing serum carotenoids, which reflect the diet in the diet on the left, to matched brain tissue on the right, the investigators found that lutein accounts for 35% of brain carotenoids, despite making up only 20% of carotenoids in serum, suggesting that there is preferential uptake of lutein in the brain compared to other major carotenoids. The study also found that brain lutein levels showed the most consistent relationship with better cognition compared to the other carotenoids. Now, taking it further to the relationship with cognition, a number of RCTs in older and young adults showed that lutein and zeaxanthin supplementation results in improvements in several aspects of cognition, ranging from cognitive fundamentals like processing speed to higher order executive functions such as verbal fluency and memory. For example, the study by Dr. Johnson and colleagues found that 4-month supplementation with lutein only or DHA and lutein improved verbal fluency scores and memory scores. So as I showed you, most of the evidence support a role for lutein and zeaxanthin in cognitive health in adults. But less is known about their role in early life, which is another vulnerable period for the brain, as I showed in the initial slides. There is some indication that lends support to a role in early life, which I'll present next. So the deposition of lutein and zeaxanthin in the body begins early during prenatal development, when lutein and zeaxanthin are transferred to the fetus via the placenta, as shown by cord blood concentrations being correlated with maternal plasma concentrations. There also seems to be preferential uptake of lutein and zeaxanthin into the breast milk compared to other carotenoids. Now, since lutein and zeaxanthin is obtained from the diet alone, maternal diet and breast milk are the main fetal and infant sources of lutein and zeaxanthin prior to the introduction of solid foods. Now, lutein has also been shown to be the predominant carotenoid in infant brains. In this study, investigators determined carotenoid concentrations in infant brains from donated samples of otherwise healthy infants who died in their first year of life. They found that lutein concentration was significantly higher, as you can see in the graph, than the other carotenoids, which are usually dominant in the diet. Now, unfortunately, the study didn't have any dietary data, but if we look at the NHANES data to see what infants were eating during that time, we see that lutein and zeaxanthin accounts for around 59% of brain carotenoids versus 12% of lutein and zeaxanthin consumed in the diet according to NHANES, which suggests preferential uptake of lutein in the brain in the brain of infants, similar to what we found in older brains. So when we compare centenarian brains to infant brains, we see that lutein is a major carotenoid in both, but the relative contribution of lutein to total carotenoids in infant brains seems to be approximately twice as that found in older brains, suggesting that there is a need for lutein during neurodevelopment. And the relationship of lutein and zeaxanthin with cognition in early life started gaining attention in the last couple of years. Here are 6 observational studies that looked at different cognitive domains among children ranging from 6 months to 13 years old. Now, the last 4 studies in the table that assessed lutein zeaxanthin status by macular pigment optical density all found associations with measures of academic performance, memory, and executive function. The first study by Mulder et al. Found no association between lutein and zeaxanthin intake with language and IQ. And the second study in infants found that lutein in breast milk was associated with better recognition memory. A recent randomized controlled trial among 5 to 12 years old, where they gave 1 gummy every morning containing either lutein and zeaxanthin or placebo for 180 days, results showed an increase in macular pigment optical density, an increase in serum concentrations. Results also showed an increase in visual processing speed, attention, concentration, memory, and learning in the intervention group compared to the placebo group. Now, I'm gonna summarize a little bit. This is an overview of what we know so far and the gap that we wanted to fill. So lutein and zeaxanthin are the only carotenoids present in the macula as the macular pigment. Lutein and zeaxanthin may protect against age-related macular degeneration. Macular pigment optical density is a biomarker of brain lutein and zeaxanthin. It was found to be related to cognition in older adults. Lutein is the predominant carotenoid in the brain and is uniquely associated with cognition among carotenoids. Now, in early life, more research is needed on the role of lutein and zeaxanthin, but there are some observations that indicate a role in early neurodevelopment. So lutein is the predominant in infant brain compared to other carotenoids. There's preferential uptake in breast milk. The deposition of lutein and zeaxanthin in human retina and brain occur early in life. There is limited evidence that macular pigment optical density is related to cognition in children. Maternal levels intake are correlated with infant levels, and macular pigment can be modified by the diet. So all of these clues led us to the main objective of evaluating the extent to which maternal and child lutein and zeaxanthin was associated with cognition. We evaluated this objective through 3 main aims. The first one is to examine associations of maternal lutein and zeaxanthin during pregnancy with measures of offspring cognition during childhood. The second one is to examine associations of lutein and zeaxanthin in early childhood with the same measures of cognition. And the last aim was to examine associations of maternal diet quality during pregnancy with the same measures of cognition. And I've cited the papers under each aim. So to address these aims, we used data from Project Viva. Project Viva is a longitudinal pre-birth cohort. It was established with the aim to examine associations of prenatal factors with maternal and child health outcomes. Pregnant women were recruited in around 2002 during their initial pediatric visit at practices in Eastern Massachusetts. Women were eligible for enrollment if they were less than 22 weeks pregnant, if they were planning to receive prenatal care at one of the selected practices, if they were able to answer the questionnaires in English. They were not eligible if they had multiple gestation, if they planned to terminate the pregnancy, or they planned to move away from the study area before delivery. Now, here's an overview of the measures that were collected by the study. The team recruited 2,670 pregnant women. After exclusions and loss to follow-up, 2,128 mother-child pairs were still enrolled at delivery and had live birth. The early pregnancy visit was done at enrollment around 10 weeks of gestation, and this is when the first maternal food frequency questionnaire was administered. This is a food frequency questionnaire slightly modified for pregnancy from the validated Harvard FFQ, and it was calibrated for use in pregnancy. The mid-pregnancy visit was done at the time of the glucose challenge test, around 28 weeks. And this is when the second food frequency questionnaire was administered. The delivery visit was done during the birth admission in the hospital. Investigators then conducted in-person visits with the mothers and their children in infancy, around 6 months, early childhood, around 3 years, and in mid-childhood. And this is when they conducted the cognitive assessments and collected additional information. Now, Project Viva administered a variety of standardized neuropsychological tests to assess different aspects of cognition in infancy, early childhood, and mid-childhood. So in infancy, infants, when they reached 6 months, cognitive testing was done using the visual paired comparison task. So the child was presented with a previously seen photo and a new photo, and the investigators calculated how much time they spent on the new photo. This shows how much they were able to integrate the new information, which is what we call vision recognition memory. In early childhood, the Peabody Picture Vocabulary Test was administered. This measures receptive vocabulary for standard English. And then the wide range assessment of visual motor ability was also administered to measure visual motor measures. That's things like hand-eye coordination, coloring, writing, kicking a ball. These are all correlated with reading and writing abilities. In mid-childhood, the Kaufman Brief Intelligence Test was administered. This is used to measure verbal and nonverbal intelligence, and it's usually correlated with IQ. The WRAML, Wide Range Assessment of Memory and Learning, also assesses immediate and delayed memory. And then finally, the investigators administered the brief and the SDQ questionnaire. These were administered to the parents and the teachers of the children to assess executive function and socio-emotional development. Now, going over the methods, we conducted multivariable linear regressions. The outcomes were all of these cognitive test scores, which were age and sex standardized, and they were analyzed as continuous. The exposures were maternal dietary intake of lutein and zeaxanthin or diet quality, and they were analyzed separately in the first and the second trimester. We adjusted the dietary intake for total energy intake, and we categorized these exposures. In terms of covariates, we had a list of a priori confounders for these associations, and we selected covariates to adjust for based on these criteria. So we included in the models for maternal exposures, characteristics that were associated with maternal exposure in first or second trimester, those that were associated with at least one child cognitive outcome, and if they were measured before the exposure. Now, we were cautious about post-exposure measures. For example, we did not adjust for gestational age, birth weight, and breastfeeding, because these are possible mediators. Similarly, for maternal cognition and for the score to measure the environment in the home. We did not adjust for them. We adjusted for them in secondary analyses, but not in the main analyses. For the models with child lutein and zeaxanthin exposure, we adjusted for maternal and child characteristics that were associated with at least one child cognitive outcome. Now, just moving over to the results, and just to give you an idea of the sample we are working with, these are women that are, the majority of which were white, were college educated, 60% had a normal pre-pregnancy BMI. They reported never smoking before, and this was their almost half of them already had a child before, and this is their second or third pregnancy. The mean age at enrollment was around 32 years old, and lutein and zeaxanthin intake was around 2.6 milligrams per day. Children, they were half male. Most of them were born full term. 27% were exclusively breastfed at 6 months, and another 27% were receiving both breast milk and infant formula. And the mean lutein and zeaxanthin intake during early childhood, that's around 3 years, was 1 milligrams per day. So, looking at the first aim, that's associations between maternal intake of lutein and zeaxanthin during pregnancy in relation to child cognitive and behavioral outcomes, while adjusting for sociodemographic, lifestyle, and dietary factors. And for the sake of time during this presentation, I'm only going to show you the results where we found appreciable trends. You've seen all of the tests that were done, and I'll show the results where we found some trends. So this table presents adjusted differences in mean cognitive scores between each category of lutein and zeaxanthin intake versus the lowest category. Now, the very first result, which is vision recognition memory in infancy, did not support our hypothesis. So we hypothesized that lutein would be beneficial. However, what we observed was an inverse relationship between maternal lutein and zeaxanthin in the first and second trimester and vision recognition memory in infants. Additional adjustment for maternal cognition in the second model did not change the results. So this is a result that we didn't know how to explain and deserves further investigation. Looking at the cognitive tests in mid-childhood, so that's around 7-8 years old, higher maternal lutein and zeaxanthin intake during the first and second trimester were associated with higher scores of KBI, Kaufman Brief Intelligence Test, indicating better verbal intelligence. In secondary analyses, that's in Model 2, additional adjustment for maternal cognition and the home score attenuated the results, but the same trends persisted. Looking at the behavioral test scores, that's the brief test, higher maternal lutein and zeaxanthin during both trimesters were associated with lower brief scores, indicating better behavioral regulation. So that's a subscale of executive function, which reflects the ability of the child to modulate their emotions and behaviors. And after adjustment for the maternal cognition and the home score, the trends slightly attenuated, but still persisted in the same direction. We didn't see any appreciable trends with the other cognitive tests. That's for the first aim. Second aim, we looked at the child lutein and zeaxanthin intake in relation to child cognitive and behavioral outcomes while adjusting for maternal and child characteristics. And we found that higher intake of lutein and zeaxanthin in early childhood was associated with higher Peabody vocabulary test, indicating better verbal intelligence. We also saw that higher intake was associated with lower global brief scores, indicating better overall executive function. Looking at the third aim, we wanted to assess the overall quality of the maternal diet in relation to child cognition. Now, the Mediterranean diet is one of the most nutritionally adequate diets. It was demonstrated to substantially increase lutein status and was associated in previous studies with slower age-related macular cognitive decline. So we used it as an indicator for diet quality during pregnancy. We computed a modified version of the Mediterranean diet score for pregnancy, where we excluded the alcohol component, and we used the cutoffs as the median intakes of the food groups associated with the traditional Mediterranean diet, because this is not a population that usually consumes a Mediterranean diet. For components presumed, so to calculate the score, for components presumed to be beneficial, mothers with consumption higher than the cutoff were assigned 1 point. For components presumed to be detrimental, mothers with consumption less than the cutoff were assigned 1 point. So their score ranged from 0 to 9. The mean scores were 4.9 and 4.8 in the first and second trimester. And we analyzed it in 3 categories: low, middle, and high. Here are the results. We observed that a higher Mediterranean dietary score modified for pregnancy during the first and the second trimester was associated with higher KBIT nonverbal and verbal scores, indicating better nonverbal and verbal intelligence. And even after adjustment for the maternal cognition score, the association was attenuated, but the trends persisted, especially for scores for the exposures in the first trimester. And lastly, we observed higher maternal Mediterranean diet score in the first trimester was associated with lower brief metacognition index, indicating fewer metacognition problems. That's a subscale of executive function, which reflects the child's ability to initiate, plan, organize, think of the future, problem solve, etc. Now here's a quick summary of the results for the 3 studies. So in a large US cohort of mother-child pairs, exposure to higher dietary lutein and zeaxanthin, both in utero and in early childhood, may influence several aspects of neurodevelopment. First, higher maternal intake of lutein and zeaxanthin was associated with poorer visual recognition memory in infancy, and better verbal intelligence and behavior regulation ability in mid-childhood. The child's own diet also appeared to be associated with cognition at the same point in time and in mid-childhood. So higher early childhood intake was associated with better verbal ability in early childhood and better executive function. And as dietary carotenoids are a hallmark of overall healthier diet, we examined the overall diet quality by assessing adherence to a Mediterranean-like dietary pattern. And we found that greater adherence to this dietary pattern was associated with better verbal and nonverbal intelligence and fewer metacognition problems in mid-childhood. So moving on to our conclusions. So our findings, along with existing evidence from older adults, suggest that lutein and zeaxanthin and dietary patterns that provide the major sources of lutein and zeaxanthin may play a role in cognitive function at both extremes of the lifespan, so older age and early life. We think that the magnitude of the associations that we found were not trivial, because the magnitude of the association of maternal and lutein and zeaxanthin intake, as well as the Mediterranean diet score with the KBIT verbal test, the magnitude was similar to that observed in the same study between children who were ever breastfed and children who were never breastfed in the same cohort. So we think the magnitude of these associations are kind of meaningful. Currently, there are no dietary recommendations for lutein and zeaxanthin. Until efficacy and safety of supplements can be determined, the recommendation is diets high in fruits and vegetables, especially that mothers and children in most of the places consume low amounts of green leafy vegetables, which are the main sources of lutein and zeaxanthin. Finally, some ideas for future directions, and I'd also love to hear thoughts from you too, suggestions on maybe what to do next. So in Project Viva, it would be interesting to follow up on these associations and look at whether the associations continue into adolescence. So these children were also followed up into adolescence. Would be interesting to see if the associations persist. Would also be interesting to look at interactions of lutein and zeaxanthin with DHA and choline, which are also been shown to affect cognition. It would be also interesting to look at the interplay of intake of lutein and zeaxanthin with breastfeeding, formula feeding, and the child intake on neurodevelopment. Would also be good to look at the same associations, but with maternal serum lutein and zeaxanthin, which are more accurate measures of intake than self-reported dietary intake. Then it would be also interesting to look at these associations in other populations with different characteristics. So Project Viva gave us an opportunity to investigate these associations in well-nourished mothers who have good access to prenatal care. It would be interesting to see if these associations also replicate under different conditions. Also, the next step would be a controlled supplementation trial in children and in pregnant women. Finally, the mechanisms of action of lutein and zeaxanthin are still not clear. It's hypothesized that they act via their antioxidant, anti-inflammatory properties, and they help in stabilizing membranes in the nervous system. So this is also another area that deserves further investigation. Finally, I just would like to acknowledge the research collaborators on this work from Tufts University, the Human Nutrition Research Center on Aging, as well as investigators in Project Viva over at Harvard Medical School, as well as partial funding from Chem Foods. That's all from my side. Back to you, Lynn.
Well, Dr. Haya, thank you very much. This is a fascinating seminar. I think at this point, it's time to find out if the audience has any questions. So everything is clear as mud. Okay, well, I will start you off. You mentioned in one of your slides that the lutein in breast milk seemed to be more available than the lutein in infant formula. Can you mention why that might be?
Yes. Yes, there's been studies showing that lutein in breast milk is more bioavailable. Well, there are multiple factors that affect bioavailability of lutein. Most importantly, the nature of the food matrix and in which they are delivered, as well as the amount and the nature of fat that's being consumed along with it. For breast milk, it seems that the composition of the fats in the breast milk is what's making it more bioavailable to absorption. I've read a study about, for example, the enzymes lipases in the breast milk helping with the absorption of lutein. This is a unique aspect of breast milk that's not found in infant formula, which could help with the higher bioavailability from breast milk. So it's mainly it seems to be the fat content in the breast milk that's contributing to the higher bioavailability.
Okay, then you had a table in there about kale and spinach that were like 25 and 29. And then down at the bottom were avocado and egg, like 1/30th. So is it the fat in those compounds that helps the lutein be absorbed? Is that the point?
Yes, yes, that's the point. I'm trying to go back to the slide, but it's not letting me. Sorry. Yes. Yes, so even though the quantity of lutein is lower in avocados and in egg yolk, their bioavailability is higher because of the fat in the avocados, so the monounsaturated fatty acids in the avocados. The saturated fat and the cholesterol in the egg yolk seem to help with the absorption of lutein, which is a fat-soluble pigment.
All right. So does this mean that we should not be using fat-free salad dressings?
I think so, yes. We do need some kind of fat to be able to absorb all of the good things that come from these vegetables.
Okay. Okay, audience, at this point, you should probably have a question.
Or thoughts or comments or suggestions.
Okay, here's
Happy
the
to hear
question.
them.
Are there any possible interactions with other dietary compounds that may affect the bioavailability of lutein and zeaxanthin?
Yes, there's been some studies on DHA and choline showing that the bioavailability is higher. Here, for example, there's a study, I'm showing a study by Dr. Johnson and colleagues where they supplemented with lutein and then DHA and then DHA plus lutein. They found better, more effects on the cognitive outcomes in the combination groups, which indicates that there might be some interaction going on here.
Right. Now that slide brings up another question. It seems like that it's a matter of days to weeks where you can increase your macula after you've been eating these compounds. Does that mean you have to keep eating this stuff the rest of your life?
Sorry, Lynn, can you repeat the question?
Okay. Well, in a matter of days to weeks, you seem to be able to increase the content in the back of your eye, the macula, which means that you can't just take a slug and then wait a few months. You have to constantly be doing this. It's a very short-term thing. You eat it and then it'll be deposited in the back of the eye.
Yes. Yes.
And so your whole life, you have to be sure to have fruits and vegetables. You can't just do it every now and again.
Yes. Yes. Fruits and vegetables, both extremes of the lifespan, at older age and then in younger age. And it's better if you start earlier because the deposition starts earlier.
All right, we have another question. Can you please discuss further how nutrient deficiencies earlier in pregnancy can be compensated for in later time? Now, this might depend on what the deficiency is, obviously.
Yes. Please discuss how nutrient deficiencies early in pregnancy may be compensated for in later time. That's a great question, Nagla, and it deserves another whole webinar with it to answer it. In terms of lutein and zeaxanthin, I'm not sure we can't say we're talking about nutrient deficiencies yet, because first of all, I won't call it a nutrient because it's still not essential. It doesn't have a required intake. So I don't think we can talk about nutrient deficiencies at this point yet with lutein and zeaxanthin. In terms of other nutrient deficiencies, It depends on multiple factors. It depends on when the nutrient deficiency happened. Was it during a critical sensitive period during pregnancy, or was it in a period was it in a period where the organ was forming, or was it in a period where nutrient stores were just being deposited, or the organ was just growing? So different factors will determine what's the effect of the nutrient deficiency and how it can be compensated for in later life and time.
All right. Any other thoughts from the audience? Okay, I'd like to go back to a slide. You talked about the intake in Jordan, that there are several infants that don't have any fruits and vegetables the prior day.
Yes.
That was children 6 to 23 months, right?
Yes.
So once they're no longer having exclusive breastfeeding, this implies there should be some intake on a daily basis as part of complementary food, right?
Yes. And of course, cooked for appropriately to the age of the baby.
And do we have any idea why this number would be so low?
The intake of fruits and vegetables?
Yeah, exactly. Is that usual in the US? Do we have any idea? Is this?
I know that the intake is low in the US as well.
Okay.
I don't remember the exact number. I'm thinking of why could this be? I don't know. Maybe people with experience with this can help. Is it the taste of the fruits and vegetables? Is it the availability of these foods to the family? Is it that they start with more sugary items, which then leads to children not liking fruits and vegetables? Is it the parents not being aware about the importance of this in early life? Maybe.
Dr. Shabani, do you have any thoughts on this based on the demographic health survey?
I have.
Yeah, thank you. Sorry. Yeah, I think it's a combination of everything that Haya has just outlined. And I think it's really the introduction of unhealthy foods very early displaces the healthy foods. So I think the DHS that's been just done, which I think the data is just coming out, probably will give more insights on what kids are consuming in Jordan. Because they're definitely not consuming a very diverse diet at that age, and certainly no fruits and vegetables.
Okay.
I think, Lynn, there are two questions in the
Yes.
chat box.
Yes. Do you do you think that the lutein and zeaxanthin as supplements for Alzheimer's and ADHD patients will improve their cognition? Are there any studies supporting this idea? Wow.
Great question, Nahla. Thank you for the question. I haven't seen any studies specifically for Alzheimer or ADHD patients. I would suspect that there might be some improvement in cognition, but since we still don't know the mechanism of action, it's hard to predict. But I'll definitely be looking out for studies specifically for Alzheimer's and ADHD patients. And this is one of the future directions that I was suggesting, looking at these associations in different populations with different characteristics. because the population Project Viva, in which we studied these associations, are very specific. They have very specific characteristics. So it's interesting to look at these associations in different types of samples.
All right. All right. We know that beta carotene in high amounts can be very detrimental. But I also heard you say that we shouldn't be taking pills of lutein and zeaxanthin. Is there any chance of overdosing by foods, do you think?
Overdosing by foods, I think there's a low risk of overdosing by foods given the content in the food and the limitations with the absorption and the bioavailability. In terms of supplements, I don't think we're at the stage yet to recommend supplements. I think we're still accumulating evidence on the benefits of lutein and zeaxanthin. Even in the observational study that we conducted, we found that lutein and zeaxanthin was associated with lower vision recognition memory, for example, in infants, which is unexpected, but deserves further investigation. So the results seem to be consistent across, but I don't think there's enough evidence to recommend supplements. So at this stage, I would say just keeping up the recommendations on increasing intake of fruits and vegetables at this stage.
All right. Any other thoughts from the audience? Anyone from Jordan in the local area? Any questions? Okay, well, I think now is the perfect time then for Dr. Haya to provide like 1 or 2 sentences of our take-home message. What are we supposed to remember after this seminar?
Yeah. Okay, so for the seminar, I think you should remember that lutein and zeaxanthin in your fruits and vegetables are very important, mainly green leafy green leafy vegetables. So reminder to continue consuming these on a regular basis. Another takeaway message is, more general takeaway message is the importance of diet during early life. So even during during pregnancy, it seems that diet has an effect on the cognitive development of the child. So I think this is not new information, but I think it's good to remember that the importance of the diet during pregnancy, the specific food components, as well as the overall dietary pattern that's consumed starting from the first trimester.
All right. This reminds me that maybe the people online would appreciate a table of lutein and zeaxanthin content of your local foods. So you can help us with that if we can get a list of the local foods.
Yes.
All right.
Yes, that would be great.
Yes, I'm sure there's many more than that. So
yes,
that will. Okay, well, thank you very much. This has been a wonderful seminar. And I want to thank you and also all the other people who attended and made this possible.
Thank you. Thank you for the invitation. And please do email me at the end. I put my email at the end. If you have any questions, suggestions, I'm happy to continue the discussion.
Will Maha, how will people find this information if they want to see it after the fact?
Yes, so we will be posting a copy of the presentation and the webinar slides on our website. I believe my colleague Shaylin has included that link in the chat, but that will be posted on our website.
Okay, great. Okay, thank you. Bye, everybody.
Thank you. Bye.